Okay, welcome everyone. Um, the session is tools for eDNA and metagenomics, and I'm very happy that I've got the opportunity to present a number of speakers today in front of you who are going to share their current work in the field. Both eDNA and metagenomics are closely linked in one way or another, but obviously they have their own pros and cons and their application in different in answering different questions. So my name is Gibran Tahir, and I'm based in Hill Labs in New Zealand. I'm a lead bioinformatics scientist, and what we do in Hill Labs is actually around providing more services around eDNA analysis. We take comparative approaches in testing and deploying eDNA analysis methods and pipelines with automated end-to-end workflows. We also deploy long-read sequencing for whole genomes with hybrid pipelines, a lot of in-house modules for error-corrected assemblies, genomes, metagenomes, AMR, MLSTs, and phylogenies. So the idea is to help out the people who are in councils, in academics, or researchers to explore the diversity of the organisms, both in the waters and in the terrestrial system. In terms of databases, there's a huge information out there in the public domain. We also leverage those, and we also try to create our local databases to help find something that is missing from the environment that is being explored here in our part of the world. And on the computing side, we are pretty strong in IT infrastructure and cloud computing. And in terms of sequencing, we leverage both short reads and long read sequencing from Oxford Nanopore. So I'll move towards today's session and full with some bright speakers. I'm going to introduce them one by one, and then we will start listing them. So first is Amali. Amali Trimamitana. She's a senior bioinformatician working at Planet Food Research, is currently a third-year PhD student. Over the course of her career, she has participated in various research projects involving a wide range of species, including both pathogenic and beneficial microbes, insects, pest, fish, and plants. Her expertise are in bioinformatics, spans across key areas such as genomics, transcriptomics, metagenomics, and comparative studies. And she's currently focusing on microbiomes and its interaction with the host, especially the Tonga species such as Manuka, Radula, and Tenur. Then our next speaker is Rio Iwamoto. Rio is the director of Advanced Sentinel, a company specializing in environmental surveillance and monitoring solutions. Under Rio's leadership, Advanced Sentinel is pioneering innovative technologies to address pressing environmental challenges. Recently, Rio introduced the company's latest product, Quick Concentration, which aims to revolutionize environmental sampling and analysis with enhanced efficiency. A pre-print paper detailing the development and performance of this tool has already been published, showcasing the company's commitment to advancing scientific research and practical solutions in environmental monitoring. Next is Helen Thas, is a food engineer with a strong background in genomics, biotechnology, and environmental DNA research. With degrees in bioproduction and technology, food engineering, and masters in biotechnology from Ankara University, she has gained diverse experience across academic and industrial settings, including positions as a product development and validation specialist. She also worked as a laboratory coordinator at the Evolutionary Genetics Laboratory. Among their key accomplishments is the successful application of metabarcoding to determine the biological origin of the honey as part of their master thesis. This work has not only demonstrated the functionality, but also provided a reproducible workflow for similar studies. Helen has also contributed to a project called A Microbiome Perspective to Secondary Contact in Alpine Butterflies, which has carried out in the collaboration with the University of Nashville and Franchi Comte University, France. Sorry if I pronounced that wrong. She continued her studies on different topics such as identification of biodiversity in extreme climate, the effects of mucilage on biodiversity, and protocol optimization of DNA and eRNA. That's cool. And omics data integration with AI. Currently, Helen is managing a TÜBİTAK-funded project titled Metabarcoding of Airborne DNA, eDNA, in the taxonomic identification of environmental samples. Next speaker is Martin Kellens, is Martin has applied DNA-based methods throughout his research career to answer fundamental and applied questions in ecology and evolution. He obtained his PhD at KU Leuven Aquatic Biology Lab, investigating how environmental factors and host genetics shape the microbiome of the water flea, the FINA, and how the microbiome composition influences the host phenotype. He then joined the CNRS, Center for Evolutionary and Functional Ecology, as a postdoc to study molecular evolution in bacteria using both comparative genomics and experimental evolution. After a short postdoc at Ghent University on the genetics of microplastic-associated bacteria, wow, he joined the Flemish Research Institute of Agriculture, Fisheries and Food, where he currently works as a researcher. His current work focuses on metagenomics, monitoring of seafloor biota, ranging from bacteria to large animals, to investigate how human activities are influencing the ecology and evolution of communities. Next is Morgan Olmsted. Morgan received her master's degree in the biology at the University of New Hampshire in 2018. Afterwards, she worked as a research technician at Vanderbilt University Medical Center Sequencing Core in Nashville. Then with the USDA stored product unit in Manhattan. With the USDA, she helped publish insect genomes and pathogen microbiome studies of insects used as feed and food and feed. Good. Currently, she's pursuing her PhD in entomology at North Carolina State University with a focus in genetic pest management in the Lorenzen Lab. She is a geneticist and genomic scholar, as well as a genetic engineering and study fellow, and is continuing her research on pathogen surveillance in insects, sulfate genetic elements, and improving insects as a food source. And our last speaker will be Ben. Ben is originally from Chile, where he earned his bachelor and honors in biotechnology. During his undergraduate studies, he went to the University of Regina, Canada, and learned about CRISPR-Cas. And then after he applied this technology to detect harmful algal blooms, then he moved to the University of Otago to Gimmel's lab. Wow, familiar. I'm just reading from this. So he's now applying into artificial intelligence to accelerate the development and deployment of CRISPR for environmental DNA-based biosurveillance. So the group's notable work has been focused on the deployment of a variety of tools for environmental biosecurity and biosurveillance, where he has been guided and mentored by Dr. Neil Gemmell and a few others. So the idea is to develop and deploy CRISPR-Cas intelligence technologies to streamline its application in eDNA targets. That's pretty much. And I think I would like to hand over to the AV team to start the talk. Thank you.
Thank you. I'm Amali, and today I'm going to give a talk on the recovery of metagenomically assembled genomes from a plant-associated microbial communities. So metagenomically assembled genomes, or MAGs, are quite vital in the microbiome field, as it has a rich information content, including the capturing the unculturable microbes as well as getting the functional roles of these microbes they play in the community. So before I get into it, I thought I'd give a little bit of a background on my project. As part of my PhD project, I'm exploring the microbiome of the manuka plant, which is a native plant in New Zealand and is commonly associated with lots of microorganisms. And some of these microorganisms has shown to have a role in nutrient intake as well as pathogenicity. So as part of the project, we wanted to explore how the microbiome shift across the tree. So we have sampled multiple tissues and generated shotgun Illumina data and some Oxford Nanopore data. And looking at these data, we see that there's a high host contamination in the younger tissues, such as the flower, such as about 99%, compared to the mature tissues, which is around 50 to 60%. So my goal was to try to capture this microbial diversity as much as I can from this data and try to increase the number of bins I can gain from it, bins or mags. So I carried out a typical workflow where I QC the DNA data and do read taxonomic diversity, as well as carry out metagenomic assembly, where I did both core assembly as well as individual assembly, where co-assembly is where you group likely samples together and assemble, and individual assembly is per sample basis. Then carried out binning of the microbes, both looking at bacteria, viruses, and fungi. And with the number of tools out there, there's a lot, and it's new ones coming out every now and then. So in the field of short-read assembly for metagenomes, it has sort of stabilized, with the main ones being megahead and spades, with the new ones for hybrid long reads coming through. With the binning, there's a lot of binners out there for bacteria, with a few coming for viruses, and not much for fungal binning, with it being a more ad hoc basis. So, and there has been attempts to do standardization and performance reviews, such as the QIIME challenge, where they look at how sampling complexity affect the binning. So looking at my assemblies, where I assembled across the different tissues, I used megahead and spades and carried out co-assembly and individual assembly. With these assemblies, I see that spade assemblies are less fragmented. However, megahead assemblies have a higher assembly size and a higher N50. Moreover, I noticed that spades have a high memory requirement compared to megahead, and megahead is much faster. And for this talk, in terms of binning, I'll just only talk about bacterial binners, and for which I tried 5 different binning tools. So I had MaxBin, Concord, and MetaBat2 binning tools, which uses a statistical approach to do clustering, and SemiBin2 and Kanban, which has a machine learning-based approach incorporated into the pipeline prior to doing the clustering, making it more effective in binning. And as a result of binning, I carried out binning on both core assembly level as well as individual assemblies. I noticed that the neural network-based assemblers resulted in more bins compared to the other 3, although Concord still are in par with the neural network base. However, when you consolidate, the number of bins you get is low in the end. This could be because of the high host contamination that I had, as well as the micro eukaryotes being there. Moreover, with the core assembly, I was able to get a lot more bins, around 50-ish, compared to individual assembly. But when you dig deep into the completion levels of these bins or mags, I noticed that the individual assembled genomes are a lot more complete than the core assembly, some of them. And then looking at how the different tools contribute during the consolidation, I see that Kanban and MetaBat2 bins are much of the bags that's coming through, with Concord being in the middle. And how about looking at different assemblers and how it affects on the binning? In terms of megaheads, was a spade. At the short read level, not a lot of a big difference seen, but I do see a high number of bins resulting as a result of hybrid approach, showing that having the long reads in the mix is of use. So to sum up, there's no one size fit all in this scenario. The coverage and the host contamination will play a role. So when you have high host contamination, the number of bins you get might be low. Same with when you have the micro eukaryotes as well. And also your amount of compute resourcing that you have will play a role in the workflow you use. So spades is a high compute algorithm, so that you need a good enough RAM and time consuming as well. The takeaway message I would recommend is, for me, this using the multi-binning and assembly strategy have worked and have resulted in a lot more bins than just using one approach. And I can see that the deep neural network binning approach is being helpful, with Kanban coming as one of the main binners, bringing me the mags. So with that, I'll finish off by thanking my supervisors, Kim, David, and Maren, as well as bioinformatics team and Hanley Lab. Thank you.
Hello, everyone. My name is Ryo Iwamoto from Advanced Sentinel. I'm here today to introduce you to QuickConc, a novel eDNA capture method developed by our team at Advanced Sentinel in collaboration with Kobe University. Traditional eDNA concentration methods, like using glass fiber filters or cervex, faces some limitations. They are often restricted in the volume they can handle, especially with turbid water samples. They can be time-consuming and may lack the sensitivity needed for reliable detection. To overcome these challenges, we've developed a QuickConc. This innovative method significantly increases the water volume you can process, particularly in turbid condition. The workflow is very simple. You just add a glass fiber sheet and a proprietary reagent, which main component is benzalkonium chloride, and to your sample and mix vigorously, and then filter it through a mesh sheet. This is a brief image of the principle of nucleic acid concentration. Benzalkonium chloride back has its positive charge, and it promotes binding of nucleic acid and glass. The suspended solid also charge negative, so it can be also attached to the glass fiber sheet as well. The suspended solid and glass fiber sheets are filtered through a mesh sheet. This is a demonstration video of a QuickConc. As you can see in the demonstration, QuickConc is user-friendly and efficient for on-site nucleic acid concentration. The entire process is designed for ease of use and can be performed quickly in the field. Our results show that QuickConc consistently outperforms traditional methods. It yielded significantly higher total eDNA across river, sea, and pond water samples. Furthermore, in species-specific qPCR, QuickConc demonstrated a remarkable increase in sensitivity, allowing for more reliable detection of target species. Metabarcoding analysis using MyFish further supports QuickConc's effectiveness. In river water, QuickConc captured a significantly higher number of fish species compared to other methods, and its impact was also evident in the NMDS analysis. While no significant difference was observed in seawater samples, the total overall trend indicates the potential of QuickConc for diverse environmental applications. This is our summary. QuickConc offers a fast, efficient, and power-free solution for eDNA capture. It overcomes the limitations of traditional methods, enabling researchers to process larger volumes of water and achieve higher sensitivity in eDNA analysis. We are now conducting large-scale studies to further validate QuickConc's performance in various environmental settings, and also continuously improving our kit components as well. We encourage you to visit our website if you are interested in. Thank you for your attention.
I'm Pelin. I will present this project. It focuses on interaction between plant species and honeybees. DNA from honeybees can be metabarcoded to study communities, but little attention has been paid to interaction between plant species. Nutrition is an important part of the well-being of honeybees, for which studies are ongoing on how they choose nectar and pollen sources. In addition to the effect of early detection or detection of unwanted species, the use of high-efficiency tool is becoming widespread to accurate applications in terms of the health and traceability of the hives. By using metabarcoding, it's possible to obtain a lot of information such as the traceability of species diversity, flight distances, preferences, or health obtained from the intestines of individuals in the hives. In the study, you can see the general workflow. We used the guts of 7 different honeybees. We can say that we obtain high quality and clean DNA at the end of extraction. We then choose ITIS for applicant sequencing because our target organism was plants. We choose NovaSeq and SP flow cell sequencing because the product size we obtain in PCR application was approximately 400 base pair. As you can see from the bioanalyzer results, a successful library was created. A minimum of 100K readings were achieved per sample. Finally, we used various parameters in the bioinformatics section. When we look at the results, we see that there are 4 dominant species, but when other species are evaluated, we can say that they are quite common in Turkey. Here we tested whether DNA extracted from the guts of honeybees show signs of specialization that would allow us to optimize sampling strategy and provide new insight into the ecology of the environment. Tracking regional plant biodiversity throughout the plants that bees feed on provides a powerful method for observing plant diversity in both agricultural areas and natural ecosystems. This can provide new insight into plant-pollinator relationships. The pollen collected by bees from different ecosystems can be used as an important tool to identify the distribution of plant species via environmental DNA. This can be applied in areas like ecosystem, restoration, and conservation biology. Data obtained from bee gut DNA can be useful for assessing plant biodiversity in agriculture landscapes and indirectly the overall health of the ecosystem. This kind of analysis can help better understand the pollination recruitments of agriculture crops. Since studies focus on biodiversity indirectly toward bees' food source are relatively new in the literature, this research could provide innovative contributions to both plant biodiversity monitoring and pollinator ecology. You can follow me with this QR code, my PhD student, PhD study, and thank you for listening.
Okay, hello, my name is Martijn Collins. I'm a researcher at the Flanders Research Institute for Agriculture, Fisheries and Foods, and I will present to you a strategy we developed for the PCR-free DNA-based characterization of metazoan bulk samples for environmental monitoring. So I work at the Marine Biology Lab, and one of the activities we do is ecological monitoring of macrobenthos communities in the North Sea. We've been implementing metabarcoding to do this for already quite a few years. And as you can see on the picture on the left, the macrobenthos communities in the North Sea have quite a broad phylogenetic diversity. So we have cnidarians, we have sponges, we have arthropods, mollusks, echinoderms, annelids. And because of this very broad phylogenetic diversity, it's quite difficult to design universal primers which amplify barcodes efficiently for all of the species. So this causes what we call amplification bias or PCR bias. So for some species, barcodes are amplified very efficiently, which causes them to be overrepresented in the communities. And for other species, barcodes are amplified not very efficiently, which causes them to be underrepresented or even absent in our communities. And because of this, we've been looking into using PCR-free shotgun metagenomics. And after experimenting a bit with different bioinformatics approaches, we decided on using QIIME matching for a taxonomic classification. So this is a method which is commonly used in microbial ecology. And Kraken 2 is a software program which is quite popular for this, and also the one we used for our analysis. But we actually had one problem, which is the reference database. So Kraken 2 relies on assembled genomes to make a reference database, and it then indexes 35 base pair sequences of DNA. But actually, for none of the species in our community, we have assembled reference genomes. So we only have the COI meta barcode or the COI barcode. And so assembling all these genomes seemed a bit daunting to us. So as a lab, we don't have the capacity to make all these genome assemblies. And well, I was feeling a bit like the panda in this comic. And actually, the solution we came up with is very similar to the one that you can see here. So we had low coverage whole genome sequencing data or genome skimming data for some of the species. We were using this to assemble mitochondrial genomes. And well, we actually had the idea to completely circumvent genome assembly and use the unassembled reads to construct a reference database. So after writing some small scripts, we managed to do this, and to our surprise, this actually worked wonderfully well. So what you can see here in this graph was a test we did with 2 fish species for which we had reference genomes, and so we did metagenome assemblies, and what you see on the y-axis is the percentage of metagenomic reads assigned to a particular species relative to what you would get when you use a reference genome. And on the x-axis, you can see the sequencing depth we used to construct the reference database. So at 1x coverage, you can see that we already classify 60% of the reads. And if we go up to 6x genome coverage, we actually classify as many reads as you would do with using a reference genome. And this is a sequencing depth we can actually achieve for many of the species, so we can construct a reference database for all of our species in the community. When we use this method, we also see that we have we have similar diversity metrics as we would get with metabarcoding, but if we look at the relative abundance estimates, these are much better because it's a PCR-free method. So on this ordination, you can see each sample is indicated by a different color. And so each sample the composition was determined with 3 methods. So the circles are the actual relative biomass. The triangles is relative read abundance with metabarcoding, and the squares are relative read abundance with metagenomics. You can see that the metagenomic samples are always much closer to the relative biomass than the metabarcoding samples, except for these 3 samples. And these are actually samples where this sea urchin was present. So it's quite a heavy species, and so it has these heavy spines and heavy shell, but it contains, well, relatively less soft tissue, so also less DNA. So this was a bit the explanation for these 3 samples. What was also good is that we can get these results with read numbers which are very similar to metabarcoding. So what you see here is that with 1,000 reads, you actually get already quite similar results as what you would get with more than 10 million reads. So the Bray-Curtis similarity of these is always higher than 0.9. If we go up to 10,000 reads or 100,000 reads, this actually improves even more. It's also a bit dependent on the diversity in your sample. Okay, to summarize, so we can use unassembled reads from low coverage whole genome sequencing to construct a QIIME index database for taxonomic classification. Our results match those of using assembled reference genomes, but at much lower coverage. Our PCR-free method also improves the relative abundance estimates, and we can get this with the metagenomic sequencing depths, which are comparable to the ones used in metabarcoding. Okay, so I would like to thank my colleagues for contributing to this work, and well, if there's any questions, you can ask them or send me an email on my email address. Thank you.
Thank you for allowing me to give my talk today. My name is Morgan Olmsted, and my title is Know Your Host, Pathogen Detection in Filth Flies. The presentation today is based on previous work with the USDA, where we worked on how to use insects as a protein component to livestock diet, and how to preemptively make it a safe food source by screening for various pathogens. The study I am talking about today is expanded off of our paper published earlier this year, where our veterinary students use sweep nets to collect filth flies from different farms over the course of the summer. In this paper, my role was to characterize the microbiome of the flies and identify any bacterial pathogens using DNA sequencing. However, we wanted to expand this research to see what other pathogens besides bacteria may be present. So in our new study, I used both DNA and total RNA from the same samples, looking for viruses, parasites, fungi, and other kinds of pathogens. We used the host genomes from the 2 fly species, Musca domestica and Stomoxys calcitrans, and the 2 host animals from the farms to clean the reeds. But as you can see on the bar chart on the right, insects still made up the majority of overall reads, but we did identify many bacteria, viruses, and in smaller amounts fungi and parasites. For this project, I utilized 3 custom databases screened using Kraken 2. These are the tan boxes in this pipeline shown. The first contained reads from RefSeq of various taxa for general classification of the samples, and then the other 2 databases were focused on pathogens not commonly included in general databases that would be of specific interest from a filth fly perspective or specific livestock perspective. These databases came from Eukaryotic Pathogen Database, EUPathDB, spanning Cryptosporidium, Plasmodium, Toxoplasma, and more, and the PR2 database, which I used to screen for platyhelminths, cestodes, and nematodes. And externally, I also looked at these reads under the FDA's Mitocheimer module in the green box, which utilizes mitochondrial sequences and is built specifically for food safety. I don't have time to dig deep into all of the taxa I found, but I wanted to share a small side story involving the use of these databases about understanding the biology of your host system. Early on, I was using more general databases that contain parasites such as Plasmodium and Trypanosoma and other human-related pet parasites, and I was seeing some small read counts in these taxa, which is a concern if we were going to feed these back to livestock. So I dug into the sequences and found very mixed results, which were not indicative of anything concrete, and I was really confused. Well, after digging further into these parasites and their biology, specifically with the 2 flies, I discovered that these flies tend to have their own parasites, which are closely related to the ones I was identifying with Kraken in my old database. This trypanosomatid called Herpetomonas muscarum is closely related to the human parasites as seen in this phylogenetic tree, and when added to the database, basically fixed the false matches. While I am not 100% sure that just adding the genome straight into the database are the best method moving forward, I think it is valuable to dig into the host of that you're working on and think about what other taxa may be there that isn't in your current database or your initial question, because what you might think might be a scary vector of disease might actually be a less scary cousin. Thank you for listening to my talk. I would like to thank those at the USDA who made this project happen, and the vet students who collected the flies and ran the DNA extractions, and my current advisor at North Carolina State University for support. Thank you for listening to my talk.
Hello, everyone. So I'm Ben. I'm coming from the University of Otago, and today I will be presenting CRISPR artificial intelligence for environmental biosecurity. So I'm a PhD candidate in genetics, and I'm applying CRISPR-Cas-based technologies for next generation environmental biosurveillance. So a little bit about the technology is that CRISPR works fairly similar in some extent like to qPCR. So basically we have a primer which we call the guide RNA, which guides the Cas13 to the target by complementarity. And whenever it finds the target, it will then make a transcollateral activity, which will cleave an artificial reporter and that will release fluorescence. So the problem and opportunity that we have in our work is that we have the invasive species. In my PhD, we work with aquatic species, and basically this generates a lot of economical losses, but also very high cost to ecosystems. They damage the ecosystem, the sustainability, and everything. So we see an opportunity here to deploy novel technologies like CRISPR-Cas for detection from environmental DNA. So our hypothesis is, can we use actually machine learning to accelerate CRISPR-based biosurveillance? Because getting the primer correctly is very hard. And our aim is to deploy and validate this machine learning on the CRISPR-Cas13 designs. So the timeline for our methods and results is basically we design this with machine learning, then we test the cross-reactivity to check that the machine learning got it right. Then we do speed testing, then sensitivity, and then we test with real environmental DNA samples. So we use cytochrome C oxidase of unit 1 as our main target for Savella spallanzani and Undaria pinnitifera. The deep learning very quickly and very briefly detects all the windows that you can potentially make a diagnostics. Then it ranks them and it tests how this could be, how it is gonna behave for diagnostics with Cas13. So then it will provide you the full assays, fully deployed with the scoring matrix, so you can rank them. And it will give you the primers, the guide will give you everything you just need to send them to purchase, get the oligo, and start testing them. And we basically did that. The interesting part of this artificial intelligence, it can save a lot of time. And it was pre-trained with 19,000 data points, which is very interesting and it's very robust artificial intelligence pipeline that we are using from the MIT. We got this whole perspective also outside with a brief article. And this is basically what we are doing from the molecular point of view. We are running 3 reactions at the same time, RPA, T7 transcription to get RNA, and then Cas13 detection, and all of them, they are running at the same time. And we have different ways of screening this, which can be fluorescence, what we are doing right now, but you can also do a lateral flow testing, which work really similar to a pregnancy test. So our results, we basically run a full panel of cross-reactivity between species, but also potential species that could be present in the environment, just to try to get something fairly similar to environmental DNA. So this is like a whole set of different kelps, and we don't see any significant cross-reactivity, which is fairly interesting and showcase that the artificial intelligence that we're using is fairly robust to make this detection, and they are very, very specific. And we have to highlight here that we are using a huge amount of DNA that can also lead to any other problem of non-specific amplification that could potentially lead to false positive, but we don't have any of that issues here, which is fairly interesting from molecular diagnostic point of view. Then we started to do the speed testing. So, okay, we have a really good cross-reactivity, right? But what about the speed of reaction whenever we go from high quantities of DNA, which will most likely not happen in environmental DNA, to actually really, really low concentration, which could be femtomolar to attomolar in environmental DNA or even lower. And we see that we actually can detect down to 10 attomolar, which is around 5 copies per microliter. And we still have a really good detection, and we can actually make a positive call before half an hour, but we still run all of our reaction for 1 hour. And we got the same results with Undaria. We are just showing here Savella, but basically we got the same results with Undaria. And then again, we're showing Savella just because of time, but also Undaria has fairly similar results. But we are also seeing that we have some kind of semi-quantitative windows when we are seeing actually this final point fluorescence. So we see that whenever we go down into the attomolar ranges, we have different fluorescence with the picomolar ranges. So that tells us that we can actually quantify the amount of DNA, or environmental DNA, that we can potentially have in a sample. And we also did this with G-blocks, but also we did this with genomic DNA, and we got basically fairly similar results. Then we went to the real stuff, which is environmental DNA, and we did 3 major experiments. We tested environmental DNA from Undaria, where we actually know that Undaria has done an invasion and is present there actively. And then we went to another site that we know that we shouldn't have Sabella, we shouldn't have Undaria. And then we went to a site that we know that there is no Undaria, but we know that there is Sabella, and all of these sites were benchmarked with qPCR or ddPCR, so we know how the potential of presence there, and we see that they correlate. And the Undaria designs are really highly specific for Undaria, and we don't have cross-reactivity in environmental DNA samples, and the same applies for Sabella. So conclusion and future steps is that we have a really robust assay for environmental detections. We have a pipeline which really streamlines these detections and make it fairly easy to deploy for novel invasions or for conservation efforts or any other. So that's a really interesting aspect. And the future steps is that we need to provide more environmental samples testing so we can actually know way more robustly our assay and how it's behaving. But so far we have extremely promising results. So I want to thank my supervisory team, Neil Gamble, Joe Zayton, Anastasia Taiko, Jerian, Javier Pachon, Jackson Tracy, and Ula, who has helped us with the NBI, the Cochrane Institute, the University of Taiwan, the Marine Base Security Toolbox, which my project is frameworked. And also I would like to thank my family, and thank you so much for coming to see this presentation today.
Okay, thank you very much, everyone, for the talks, quite engaging. And we have all the participants, well, if not all, most of them, who can probably communicate and comment on the questions and answers. I put most of them, but there's one more from audience too. Yeah. So I think first I'll take the audience question. Hi, Pelin, great talk. Thank you. But Pelin is not here, obviously. Sorry, I may have missed the detail, but what the reference plant database that you typically use? Yeah. Pelin is unfortunately not here to answer the question. Yeah, so we can just pass on that one. I have a question for you, Amali. For the fungal genomes, I know they're harder to build. Was there a particular approach in ML that suited eukaryotic genomes over the prokaryotic microbes?
So there's no real fungal business out there currently. So what I have been doing is extracting fungal bins from the bins that I made from the bacterial business. And so far, looking at the bins result, Concord and Metabed gives more fungal bins than more high quality fungal bins than the semi bin. I haven't yet gone through the bin results. So I'm looking forward to having a look at that and see if the ML approach has effect or not. Yeah, so once I do, I could let you know. Yeah, thank you.
Cool. Thank you. The next question was to Martin. Yes, good talk, Martin. One thing that comes to my mind when I was listening is about the error rate and the low coverage of the reference data in the data that you build. How do you handle that issue?
I didn't catch the question very well. The sound was a bit off.
So if it's a low coverage, when you say it's low coverage, I'm getting that you have a very low read depth. Yes. Yeah. So when you have a low read depth and you have these errors usually in the sequencing, how do you handle those polymorphism that comes from errors in predicting the right sequence?
So I think for the low coverage, because Kraken is indexing 35 base pairs, you actually don't need to assemble. You can actually get like 115 35 base pair k-mers from one read.
K-mers, yeah, okay.
And so actually, like for polymorphisms, if we index, for example, we did tests with like single or two species communities. We can actually classify already like 95% of that data. So a reference coming from one individual and then from a community, which is from a very different area. We can already classify a lot of the data. So polymorphisms don't seem to be a big problem for classification potential. So I hope this answers the question.
Yep. If it's not a problem, then it's fine.
Yeah, no, well, it seems to work quite well for species level identification. That yeah.
Okay. Thank you. I have one more question for Morgan. So I think it's good. I think it's clever to combine the metagenomics with RNA-seq. Thinking about falsehets, where you will able to actually use texa to identify real ones from the falsehet. How do you handle the falsehet schools who will in the long run? Like, could it be possible that these falsehets are something associated with the field in reality? So do you have a database then you check with other countries too? Like, it's not the case, or
I think that's going to be overall a major issue in our field moving forward, is that your data is only good as the database that you have. So ideally, we would have every single organism in the world represented in our databases, but that's not going to be possible. So basically, my option is using the know the biology of your host very well,
Yes.
and potential things could be there and incorporate them into it. But that I think that is a very stressful thing to think about moving forward.
Yes, but do you share this with other countries too? Other food?
Yes, I mean, it's not very country specific, the databases that I used. They're
No. So these
Sure.
falsehets that you get in your study, do you share this information with others too? Like in other food chains?
I've seen
Prevalence,
I've seen there have been reports like people publishing on some of these these parasites being found in insects as food. However, I don't know if I trust them because some of the PCRs they are using, like you look back and that they can be kind of another taxonomic cousin. Yeah.
Cool. Okay. Helen is here. That's good. She replied. But I'm sharing my own database for my own study. I also work in honey. Yes, I'm also working on metabar coding. It's good. Okay, she has a problem with Zoom. The talk was good, Helen, if you're hearing us. So yeah, that's pretty much wraps the questions from my side. If anyone else from the speaker has a question with anyone else, you can ask. I think I request Ria to share the quick comp. Okay, thank you very much. Ria, if you can share that, we can test it in our environment quickly. Yeah, because we do a lot of surveillance testing of various kits now already, but we can test your one too. If it performs well, then it will be ideal to use it. Yeah. Okay. I think this is the last session for today, and see you tomorrow for the rest of the new sessions. Thank you for joining. Take care, and see you tomorrow. Bye.